Today I will be talking about single-cell mass cytometry or CyTOF, the protocol that we can use to perform a CyTOF analysis, and an example of CyTOF application in our laboratory. Single-cell mass cytometry is a variation of flow cytometry in which antibodies are labeled with heavy metal ion tags rather than fluorochromes. The core advantage of mass cytometry compared to flow cytometry is that there is almost no background noise, as each metal isotope has a distinct detection peak and doesn't overlap with other metal isotopes. Because of this lack of noise, we can simultaneously detect more than 40 markers from a single cell. In the instrument, cells stained with metal-tagged antibodies are nebulized into single-cell droplets and prepared for complete ionization in the mass spectrometer. The resulting ion cloud is enriched for heavy metal ions using a quadrupole that are then separated in a time-of-flight chamber by their mass-to-charge ratio and detected. Ion counts for each isotope directly mirror the expression of the corresponding targets. There are 8 key steps in a typical workflow of a CyTOF experiment. The initial step involves sample preparation. Cells of interest are carefully collected from tissues and cell cultures, or isolated from peripheral blood, in the case of peripheral blood mononuclear cells. Achieving an excellent single-cell suspension is crucial as it preserves cell viability and prevents clumping. An initial staining step with cisplatin is employed to evaluate cell viability, enabling the distinction between live and dead cells in the subsequent analysis. For experiments involving multiple samples, researchers may opt for barcoding. This consists in labeling cells with unique combinations of metal isotopes, enabling the pooling of samples for simultaneous analysis. Barcoding is particularly advantageous for reducing sample variability and improving experimental efficiency. Cells are then labeled with isotopically pure metal-tagged antibodies, each designed to target specific cell surface or intracellular markers. The optimization of antibody concentrations and incubation times is crucial to guarantee robust and specific binding. Following labeling, fixation is performed using, for example, paraformaldehyde, preserving the native state of cells and immobilizing metal-labeled antibodies. Permeabilization, achieved with a suitable detergent, facilitates the entry of metal isotopes into intracellular compartments. For a comprehensive understanding of cellular dynamics, intracellular staining could be incorporated after the permeabilization of cells to enable the detection of intracellular markers. The inclusion of a DNA intercalator staining step holds significant importance in discerning individual cells from debris, aggregates, and other non-single cell entities. On the other hand, in that analysis, forward scattering and side scattering are employed to identify cells. After staining and washing steps, cell counting is performed to achieve an appropriately diluted suspension. Once samples are prepared, the CyTOF instruments comes into play. The labeled and fixed cell suspension is introduced into the device, where inductively coupled plasma ionizes the metal-tagged antibodies. Time-of-flight mass spectrometry is then employed to measure the mass-to-charge ratio of ions, facilitating precise identification and quantification of individual cells. The subsequent data acquisition phase is crucial, capturing high-dimensional data for each cell. The intensity of metal isotopes is recorded, generating a comprehensive profile that forms the basis for downstream high-dimensional analysis. Transition metal carbides, nitrides and carbonitrides, MAXINs, are promising two-dimensional materials for biomedical applications. However, there is still a critical unmet need to detect and image nanomaterials within single cells and tissues while surveying high degree of information from single samples. Here we propose linked, a label-free single-cell detection strategy based on single-cell mass cytometry by time of flight, CyTOF, and ion beam imaging by time of flight, MIBITOF. In linked, we design MAXINs containing specific transition metals that can be directly identified by mass cytometry and imaging at the single-cell level. This design abrogates the need for additional chemical functionalization of the materials, thus overcoming many limitations of common imaging approaches. Moreover, we use new channels which have not previously been explored for commercial mass tag in mass cytometry. First, we tested MAXINs simultaneously on 15 primary human immune cells populations and successfully detected material uptake and single-cell interaction by CyTOF and imaging mass cytometry. We then evaluated the possible correlation between MAXIN cell interaction and the impact on cell viability, revealing the high immunocompatibility of the materials regardless of the extent of interaction. We confirmed the biocompatibility of MAXINs in terms of cytokine and chemokine production and effects on whole genome expression by RNA sequencing. In vivo biodistribution studies by CyTOF and MIBITOF using a mixture of MAXINs in mice confirmed the versatility of our detection strategy and revealed MAXIN accumulation in different organs and relative immune cell subtypes. In conclusion, we propose LINK, a new label-free single-cell detection strategy based on single-cell mass cytometry by time of flight and MIBITOF, enabling nanomaterial detection and simultaneous measurement of multiple cell and tissue features, which is expected to offer exciting new opportunities in biomedicine.